Well, good evening. Thank you everybody for joining us. This is the fourth in this special series with Professor Leona Gilbert, who's the editor of the wonderful book, Borrelia Burgdorferi Methods and Protocols. And we're delighted to have the authors of chapters 17 and 18 with us today, who'll be talking about advancing research in gestational Lyme disease. So I'll leave them to introduce themselves as they wish to before they speak. And please do put your questions and comments in the chat or the Q&A. They'll be talking for about 30 minutes, and then we'll have lots of time for exchange and discussion afterwards. So thank you very much again, Professor Gilbert and Dr. Nades and Dr. Gilliam. Thank you.
Yes, and good evening, everybody, and thank you for spending your time with us this evening and on the other side of the world in the afternoon and morning for some. So Dr. Nades and Dr. Gilliam, really, it's an honor to have you guys here, and thank you for your participation. And Dr. Nades, if you would like to have short cliff notes on your bio, and then we'll follow with Dr. Gilliam, please.
Okay. By background, I am a rheumatologist, trained at UCSD in San Diego, and last academic position as chief of rheumatology at Penn State before moving into the diagnostic industry, where I have been since 2006, currently as medical science liaison at LabCorp.
Great. Dr. Gilliam.
Yeah, my name is Laura Gilliam. I, by training, have my doctorate in molecular and cellular biology from Mount Sinai School of Medicine in New York City. Spent most of my training in public health with the CDC and New York State Department of Health in emerging infections. And then at LabCorp, serve as a medical science liaison in infectious disease, and also participate on several national committees to advance Lyme disease diagnostics. So happy to be here.
Great. I'm going to now share my screen, and I hope you guys can see our presentation here. So give me 1 second. Okay. As Jillian mentioned, we are going to discuss basically the two chapters that Dr. Nades and Dr. Gillian has helped us conceptualize. And basically this book, the motivation is because we really realized that there was knowledge gaps in regards to Borrelia Burgdorferi and the research surrounding certain items. And tonight we're going to talk about really Borrelia Burgdorferi in pregnancy. But basically this book was designed and written so that anybody can pick it up and really follow the procedures, can do in vitro, in vivo, in situ, and de novo, and even clinical investigations surrounding Borrelia Burgdorferi. And our only motivation for this was really to advance the frontier of this infectious pathogen and really do sound science in regards to this pathogen. So there's two chapters. Chapter 17, Establishing a Pregnancy Lyme Disease Biobank, was written by Dr. Nades. And I find this interesting because he has established many different biobanks at different companies over the years, Quest and LabCorp, and even the biobank that we had when I was doing, postdoc in his research group under his actions and his supervision. So this was really great for us to invite him to write this chapter. And the second chapter, chapter 18, Developing a Prospective Gestational Lyme Disease Study, we realized that there was really a gap in this knowledge. And I know with Dr. Nades' experience in working with, clinical studies involving pregnant women, especially in the B19 world, that this would be ideal for him to write this with his colleagues as well. And that's why we have Dr. Gilliam there to help us and help us write this chapter and have it involved in this Borrelia Burgdorferi Springer's book. So with these two chapters in mind, I'm going to give the floor over to Dr. Nades.
So what we wanted to do was to ask what it would take to develop a biobank for gestational Lyme disease, Lyme infections occurring during gestation. And in the other, what we would want to do in terms of a study that would use as a foundation this biobank. So the discussion today is going to be a brief introduction to Lyme disease and pregnancy and an overview in terms of its incidence and specific concerns related to pregnancy. The objectives of such a study would be to understand maternal and fetal health outcomes and to what it would take in that context to establish and maintain a biobank. So we will speak about briefly various, and Dr. Gillum will be able to go into more detail as requested, serological and molecular diagnostic methods, sample types, and processing protocols that we might use in such a study and for such a biobank. The biobank, given the population that we would be targeting, in and of itself, as any biobank, has certain structural requirements, need to define objectives and governance, but also because of the population we're targeting, really requires very careful thought and some additional considerations. Certainly, there are ethical considerations regarding any human subject study involving informed consent, privacy, and security issues around that data, particularly if one wants to do longitudinal studies, which require some form of de-identification in the setting of wanting to follow longitudinally subjects. So how do you support all this? There are examples of effective public-private partnerships, and this is important both in terms of collaboration for study, scientific and medical, but also for funding and for resourcing and access to subjects and materials. And so ultimately what we want to ask is, what is the long-term impact and future directions that this kind of study would contribute in terms of our understanding of Lyme disease, pathogenesis, and the impact of this pathogen on public health. Next slide. So many of us are familiar with Lyme disease, but when we think about incidence, it's quite variable geographically. So we know that in Europe, studies have suggested that there are 20 to 40 cases per 100,000, with higher rates in endemic areas. In the United States, the CDC reports about 30 patients submitted to them as reported, but estimate that there's almost 500,000 individuals that may have been exposed or infected. And so there's a gap in our understanding at a significant level is what is the burden of Lyme disease. And so, obviously the risk factors may vary. Lyme disease is transmitted by ticks and manifests the classic erythema migrans, the bullseye rash, fever, fatigue, joint pain, and later in the course of the disease, cardiac and neurologic manifestations for some patients. But clearly, the diagnosis is challenging, certainly because not everyone has the classic erythema migrans, bullseye rash. Individuals may not be aware that they have been bitten by a tick. They may not be aware that they have the primary erythema migrans rash. So being able to identify patients really requires a careful clinical history, careful occupational social history, activities, exposures, and constitutional symptoms, but also specific symptoms that we would associate, and a careful examination. And at times, additional studies in terms of the laboratory, not at times, but consistently in the laboratory. But the timing of that testing may be a challenge. And so early on, one may not see serologic response, for example, and post antibiotics, antibody responses may be aborted. So one needs to bring the full armamentarium of history examination, supporting laboratory testing, imaging for cardiac echocardiograms, EKGs, etc., to be able to define who our patients are. So, clearly with any number, perhaps 500,000 in the United States, cases being estimated and not being reported, many of these individuals will be women of childbearing age. And so, one wonders, what is the burden of Lyme disease in pregnancy, and what would be the risk to the fetus in terms of spontaneous abortion, stillbirth, congenital anomalies, or developmental issues going forward. So when one asks what is available, what do we know in the literature, there are limited numbers of studies on gestational Lyme disease, and most of these are case reports or small series. And we're looking at a handful on the average annually for the last many years. Next slide. So there are existing gaps in our understanding. As I said, there are few comprehensive studies, and most data comes from small case series, and this limits the generalizability of any conclusions. So there's a need for a comprehensive population-based study or studies, and this would require large-scale prospective studies that are needed to establish accurate incidence, risk factors, and outcomes. So in addressing unanswered questions in maternal and fetal health, we had proposed, if you will, a Gedanken experiment, a thought experiment, that aims to fill the knowledge gap on the effects of Borrelia infection during pregnancy and the long-term outcomes for the offspring of those pregnancies. Next slide. So when I say a Gedanken experiment, this was a term that Albert Einstein had coined, which was to describe an experiment done by thought, but not by actually doing, not putting hands on. So what we wanted to do was ask ourselves, what would be the ideal study if we were unlimited by resource, funding, personnel, method, etc. So the study was designed, as we proposed, to monitor pregnancy outcomes, including potential complications and developmental delays in the offspring. So we would follow pregnancies and follow the children of those pregnancies up until the age of 5 to assess developmental milestones and potential chronic conditions. The secondary goals would be to identify, detect Borrelia species and other tick-borne pathogens using available serologic, microscopic, bacterial culture, and molecular techniques for comprehensive pathogen detection. And in addition, by creating a biobank and having clinically characterized specimens available, we would also be able to provide investigators developing novel diagnostic methods to access materials that would be of assistance in their validation of those assays. And that would be one of the purposes of the biobank, to create that biorepository for ongoing investigation in Lyme disease and pregnancy. But also it'd be a resource, since we'll also have controls, a resource for asking questions about pregnancy in general. Next slide. So how do you go about recruiting subjects for such a study and being comprehensive and avoiding recruitment biases? We've thought that one way to do it would be to invite obstetrical practices in both endemic and non-endemic areas. So we have a non-endemic area control to participate. And we would ask all patients in that practice, invite them to participate, so that we would avoid a selection bias and only recruiting those who we suspected as having a Lyme disease exposure or infection. And we would want to do a variety of geographic regions to capture a wide range of exposure risks. And we would want to start early in pregnancy, so with the first trimester, with the first patient visit, in order to track the full course of pregnancy. Obviously, we would also welcome individuals who come to the practice later, but we would look at them in terms of perhaps a subset, those who present early versus those who present later, because those who present later may or may not have a bias in terms of why they came late, why they came early, etc. The other way that we would want to recruit would be to engage lay groups, foundations with interest in supporting Lyme disease, and using social media to recruit individuals who may not be involved directly in the obstetrical practices that we would recruit, but also encourage patients to encourage their providers to participate. So it would be an all fronts recruitment. So we would be able to compare within the study cohorts of infected versus non-infected pregnant women. We would be able to do the comparative analysis of pregnancy outcomes between these two groups, and they'll be within the same practice, if you will, in the same geographic areas. And so we would have to be cognizant always of what are the local Borrelia species that are prevalent, the socioeconomic status, access to healthcare, travel histories, is it a rural, urban, semi-rural area, etc. So there'd be a number of elements that we would pay attention to in terms of the geography and the demographics of the participants. Next slide. So as I said, we would do, every avenue in terms of recruitment to maximize enrollment and retention and to decrease selection bias. And so we would focus on a demographically diverse participant pool in order to be able to generalize the findings that we may have. Next slide. So I'm going to ask Dr. Gillum to walk us through the serologic testing on how we would confirm or look to confirm a diagnosis serologically. Laura?
Sure. So in putting together this chapter, we did focus in on those tests that are currently included in most guidelines and recommendations, certainly acknowledging that there are a lot of other research-based test methods that may be more sensitive, more specific, be able to detect earlier. But we decided to focus in on what is currently recommended. So for serological testing, we focused on the two-tier testing algorithm, which is typically an immunoassay, an ELISA format that is confirmed either by Western blot or immunoblot, or which is more recently approved in the US, a modified two-tier testing algorithm, which uses ELISA for the first step, and then two additional ELISAs for the second step. So that's what we included as far as the serological testing, but certainly there are some challenges and limitations. The serological testing, as we know, does not detect early infections as well. We have some data to show that the modified two-tiered does detect a little earlier than the traditional, but again, we'd be missing potentially some of those early infections. And then certainly we see false positives with other serological tests in pregnant women many times. And then it would be difficult with the neonatal testing to differentiate between maternal antibodies and those that are actually produced due to infection in the neonate. Then moving down to molecular and non-serological testing, which really aren't as common, again, as far as what is currently approved. We did include PCR for direct detection. Certainly some issues there as far as when viremia is present. And we also discussed microscopy and culture for further confirmation. And these are included in the US as part of the CDC guidance, and certainly in other countries too. Again, really where I think the research testing would come in is for that more accurate diagnosis, to be able to have early detection. Again, we have gaps in our current testing methods that are approved that don't always detect these early infections. So recognizing that in a research study, being able to use alternative methods for that earlier detection. And then we also talk about what would be the case definition for Lyme disease. And we took a look at a number of different guidance and around case definition and really saw a gap there. That currently the definitions that are utilized for diagnosing Lyme disease in the general population would be applied to pregnant women, fetuses, and neonates, but that there's really a need through these research studies to have more specific guidance. So again, recognizing with the diagnostic methods, there's those tests that are approved, and then there's those tests that really are needed for the research studies to give a better diagnosis. Next slide, I believe.
So the types of biospecimens collected would be maternal and cord blood, amniotic fluid, and placental tissue. And these would be collected through the pregnancy for serologic and molecular analysis. And of course, maternal blood is easy, cord blood only if indicated. Same thing with amniotic fluid and placental tissue in terms of chorionic villus biopsy. So, we would routinely collect maternal blood and cord blood at birth, and the other samples we would collect as additional samples for research if there is clinical need to do an amniocentesis or to sample placental tissue. One thing that is always integral for a large study with multiple sites is how do you ensure uniform processing and storage? And so it would be incumbent upon the study and the biobank to establish standard protocols for processing and storage to ensure sample integrity and standardization. And so it's not just how the samples are collected, but how they're processed on site, if they are processed on site, how they are shipped, and it's important to monitor the shipping. Shipping a specimen in Georgia in the midsummer can be problematic because of heat. So again, ice packs, dry ice, depending on what's appropriate for that sample. And to be able to track those so that they don't get lost in the transportation. And so these are critical questions for any laboratory, diagnostic laboratory, research laboratory, or biobank in terms of getting samples that in which you maintain integrity. So the samples then are stored in a biobank for current and future research initiatives. And so, there is a need for protocols, which we'll discuss later about how those samples are used, what is tested initially, what is saved for testing later, and who has access to those samples. So in addressing the logistical issues and ensuring high quality samples, that's the challenge. And so the study needs to develop solutions for timely collection and transport, and particularly from remote areas. So there are areas where individuals may not be able to travel to a provider on a regular basis. Obviously, if they're in an obstetrical practice, you would hope that the pregnant subject would be able to get to see their provider. But even so, remote areas may have transportation logistic challenges that have to be addressed and overcome. Next slide. So in establishing the biobank, the objectives are to support the study and the broader research community and provide high quality biospecimens for Lyme research and related studies. So there needs to be consideration for long-term storage and management of the biospecimens. One needs to make sure they are preserved under optimal conditions for extended research use. And one needs to maintain the operations of a biobank and to ensure continued funding in order to maintain it over many years. So that requires a governance and oversight structure that typically would start with an executive committee that would consist of committee chair, a vice chair, because succession planning is always important to maintain the integrity of a resource that one expects to be there long term. So you have a chair in waiting, if you will, in training, a secretary to maintain a recorder for meetings because meetings are, essentially need to be recorded. Many states will have sunshine laws that require, particularly depending on how this is funded, if it's funded by public monies. Also, one needs an individual who can oversee the operations and be able to report back to the executive committee how the day-to-day operations are run, as well as individuals who are responsible for supporting financing and maintaining financial support. And so there are a number of committees that support the activities of an executive committee. So subcommittees would manage the biobank operations, data security, ethical compliance, oversight of distance sites to make sure they're in compliance and so on. So there's a need to create policies for access to samples and the data, clear guidelines who can access the biobank's resources and under what conditions, both in terms of the wider data and in terms of accessing the materials. Many biobanks will utilize a scientific review committee that will vet proposals for support of outside research projects. And so one needs to maintain that rigor and balance on that kind of program to make sure that there is an open mind to what studies will be resourced and under what conditions. And so obviously, none of this works without strict adherence to ethical guidelines and the use of samples for research and protection of human subjects. Next slide. So the informed consent process is a requirement. And typically when you have a large multi-center study, central IRBs, internal review boards, are used. But in institutions that are participating that have their own IRBs, the local IRBs will also want to review the protocols. And there's particular sensitivity and attention to what are considered vulnerable populations of subjects. And these include pregnant women, fetuses, and neonates. So one has to be very careful about dotting I's and crossing T's in order to maintain approval and to protect those participants who have volunteered to engage in the study. The data that is collected must be handled under HIPAA in the United States and comparable laws in other parts of the world. So the Health Improvement, Portability, and Access Act and other relevant regulations to protect participant privacy. So that requires a great deal of protection for that data, how to de-identify the data, and yet provide a code that at the provider level would link a patient's visit from first trimester all the way through third trimester and all the way through for fetuses up to 5 years to be able to follow that as a data set and yet secure the identity and de-identify the individuals in the biobank. So you need to be able to track samples. And so one needs to create a firewall between the provider and the subject, the provider needing to know the data and de-identification. The other issue that one has to deal with in this kind of study where you're actually doing diagnostic testing, one has to ask the question, is that data going to be provided back to the provider and the patient? If so, that data, if it's going to be clinically used, needs to be performed in a CLIA, Clinical Laboratory Improvement Act, approved and licensed laboratory, whether it be an FDA approved test or a laboratory developed test, or even a research use only test if it's being provided to the patient, needs to be done under a CLIA umbrella. So one needs to make that decision as to what is going to be provided back and how to do that in a way that's appropriate. So these protocols need to be maintained for de-identification to provide for ensure confidentiality, but also to provide access for information that may be relevant clinically in real time to practitioners. And so that's a careful balancing that one needs to do. The IRB I mentioned, regular IRB reviews to ensure ethical conduct, and this is typically on an annual basis. So administratively, there's an administrative burden to running a large biobank, but it's important in order to maintain the integrity of the biobank and address ethical issues. These need to be carefully thought out and approved by IRBs. Next slide. So how do you support all this? Well, there are very good examples of public-private partnerships for funding and resourcing. There are any number of resources, both in terms of funding, testing, expertise, and the one very successful example we can point to is the Alzheimer's Disease Neuroimaging Initiative, which is funded by the NIH Foundation, individual pharma companies, individual foundations. And the model has been very effective in collecting patients with Alzheimer's disease, their neuroimaging, biospecimens for testing serum and CSF. And there's a central statistical core, there's a central laboratory core, and a central IT core that collects this data. And for participants in the study, as one example of how to manage data utilization, the data is available to individual participants who are submitting, and their data is available only to them for the first 9 months. At least the last time I looked, it was 9 months, after which de-identified data is available to all participants. So it's been a very effective model in the neurology space. So we can look for this kind of study to an array of potential partners. The NIH, traditional NIH funding mechanisms, NIH Foundation, the CDC, pharma companies, diagnostic laboratories, national diagnostic laboratories, for example, like ours, and foundations who have an interest in this area and partner and create partnerships to move this forward. And to build and sustain those partnerships, you need clear goals, regular communication, mutually beneficial frameworks to maintain those partnerships, and a lot of interaction. Next slide. So, the potential impact of such a study is to establish new guidelines for the diagnosis and treatment of Lyme disease in pregnancy, and to develop new diagnostic tools and treatment. Certainly, we would have clinically characterized specimens in order to look at biomarkers of infection and biomarkers for disease outcomes for morbidity and mortality. So this could also, ultimately lead to new therapeutic approaches on how to manage women in their pregnancy and how to manage the offspring, the fetus, and the children that are born. So once you have this resource, the biobank can certainly look at research projects beyond just Borrelia, certainly basic questions about pregnancy, but also other pathogens, Babesia, Leptospora, for example, anaplasmosis. There are other tick-borne pathogens that one can consider evaluating in this same patient material. And the long-term value to the scientific community is immeasurable because it's a resource for ongoing and future research. And, the heavy lift is in gathering this data and these materials. And so any number of innovative um, seed projects can be spun out from this material. And so we look for potential opportunities to leverage the biobank and the study data, looking at co-infections, long-term outcomes, and new diagnostic markers, and enhancing collaboration across research disciplines, because cross-disciplinary studies could yield perhaps innovative approaches and insights to Lyme disease and pregnancy. Next slide. So in conclusion, and for greater details, I would refer listeners to what we wrote in the text. This would be a comprehensive approach to studying gestational Lyme disease and would be able to integrate prospective study design with a biobank establishment. And clearly the biobank has a great deal of importance for future research in our ability to advance our understanding and treatment of Lyme disease. And so as a final thought, we would call to action for continued support and collaboration, encourage partnerships and funding to ensure study success, and certainly emphasize the impact on public health and highlight the potential for significant public health benefits. Next slide.
Great. Oh, final words, Dan?
Well, I was just, you had
Go ahead.
also brought up, there are some biobank initiatives currently underway,
Yeah.
and
Mhm.
I guess Dr. Gilbert will point those out.
Yeah, I just wanted to just mention the active ones right now that are recruiting individuals is these two that I was able to find at the clinicaltrials.gov website, as you can see here, the website address. So the first one is the pregnancy and early neurodevelopment outcomes following in utero Lyme disease exposure. And then the second one was navigating pregnancy and parenthood with Lyme disease. What's interesting about these two research projects, they are run by the same people and funded by the same people and have the same collaborators. So the Children's National Research Institute. And it's interesting that these are the two most current ones, but I also know that there's some really great resources on children in Lyme and also pregnant women in Lyme, some resources that you can go to at this website, LymeDiseaseAssociation.org. They have a good listing of collaborators there and good listing of resources that you can find out more information. And usually they're on top of the kind of like the news outlet of what's going on in the world in regards to pregnancy and Lyme disease. So I just wanted to mention this for you, and also really, as Dr. Nate said, call to action to get involved. If you have, if you have patients that are pregnant, it would be really great to try to get them enrolled. At the moment, both of these projects, the first one has, I think, 40 individuals enrolled, and the second project has 30. So not too many, but again, hoping that their recruitment is going on longer. And both of these studies are supposed to end next year, 2025. So I'm not sure how long the recruitment process will be open, but it's still apparently open for both of these. So I just wanted to give you that information and really thank Dr. Nates and Dr. Gilliam for being here. And also to look out for how you could participate in, establishing a biobank and also establishing research kind of initiatives, because it takes, healthcare providers, it takes scientists, it takes stakeholders, it takes the patients, to get involved in these types of initiatives. And more and more research projects demand the public patient involvement in anything that we do. And obviously we cannot do any of this research in these two book chapters if we didn't have, the pregnant woman and other, availability of biospecimens and so forth.
Yeah. So
And
one,
yeah,
let
go ahead.
me just add in that context,
Yep.
one of the representative groups or committees that one would have in a biobank such as this is a patient participation. It's very typical now of many such resources that there are patient representatives providing input, and that's discussed further in the chapter.
Yeah, and that's an excellent point, Dr. Nates, because I just wanted to also mention even funding schemes like the European Commission demands it now for research projects that are seeking their funding to really have patient public involvement in everything that research project is doing. So it's really important that we consider them from the onset. Okay, and then I wanted to provide you, because I always harp about this in all of these roundtable discussions and also all my lectures, is that publish, 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 publish. I encourage healthcare providers, I encourage scientists, and encourage the patients too, if they're involved in research, to get involved in publishing. And one of the ways that we can help with that is actually directing some of these articles in the research to these kind of very focused kind of special topics. And the special topic that's open with my collaborators, like Dr. Lambert and Dr. Sean and Dr. Sappi, is this diagnosis and treatment strategies of tick-borne diseases in frontiers microbiology, as well as frontiers in cellular infection microbiology, also frontiers in public health and frontiers in medicine. And I just wanted to point this out because we have the goal with our colleagues to actually provide a friendly place for submission of research that undergoes a rigorous peer review process. And if you are interested, please contact me, or you can also go directly to Frontiers Microbiology and see this really focused special issue.
Fantastic. Thank you so much, all of you. By the way, Leona, when you say they should contact you direct, would you like to put into the chat an address
Sure.
that they can contact? That would be
Yeah.
excellent. Thank you.
Yeah.
Would probably be best direct to you rather than to the journal itself, I can imagine. And of course, anybody who'd like to volunteer their patients or even themselves for studies, do either look at those two that are ongoing or contact@aunm.org would be a place where we could collect that data and then send it on. Do you feel, just because you mentioned those studies a moment ago, Leona, that the numbers are sufficient, or would you ideally look for quite a few more participants in your cohort?
Yeah, I would say that we need more, but I know it's difficult to get, these controls. Like, we need controls, and as Dr. Nates mentioned, we need individuals from endemic, non-endemic areas. We need patients that are not pregnant, so we can compare them with the individuals that are pregnant. So we need more enrollment. And statistically speaking, if we try to enroll like 50, minimum 50 participants in each of those categories, that could be a robust kind of clinical trial. Or what do you think, Dr. Nates? I know it's difficult to get 50, but
I think for statistical purposes, I think in big numbers.
yeah,
you consider the number of suspected unreported cases. There's a large population out there that may be exposed. And I would want, at a minimum, hundreds, if not thousands, of participants in such a study. I mean, as I said, this was what we proposed was a Gedanken experiment. It was a thought experiment. What would be ideal?
Yeah.
There are real challenges to doing this kind of study, both in terms of time, personnel, resources, storage. Every step of the way, there's a challenge, but it's not something. We put a man on the moon. Theoretically, we could do this. The question is there a will? Is there funding? And are all the elements that are required to be successful being brought together? And so that's the element. I mean, to really draw conclusions about risks in a general population, in a general pregnancy population, you need a lot of subjects. And so think big. Now, what I've suggested, again, at the end of the chapter is, personal thoughts. I mean, when you think about a biobank, if you're thinking about starting a biobank, you think small, but you plan big. And so you build on your success. And so the more subjects, the better. The data becomes stronger if you have, bigger pool.
Well, thank you so much. That was a fascinating presentation and so systematic, a wonderful framework that you've presented. And I'd just like to go through some of the questions here. I'll go through them from the top, as it were. A lot have come in. So one participant says, well, she recognizes the gaps that you have also mentioned in Lyme disease testing. She says, if co-infections such as Babesia, Bartonella, and others that can be gestationally transmitted were also tested for, the potential for one of these yielding a positive would increase, and therefore a tick bite could be suspected more easily, I suppose. Ticks everywhere are co-infected. Most people I know are co-infected and have never had an EM rash. Is that something that would be potentially conceivable as well?
Laura?
Absolutely. I think the recommendations are largely moving towards looking for co-infections, not focusing just on Lyme. So absolutely, with the samples available, I think it would be very valuable to look at those other vector-borne diseases that could also be transmitted at the same time or separately, but have similar impacts.
Yeah, and we discussed that in the chapter. I mean, this would be something we would want to do.
Super, thank you. The participant also mentions that, if possible, case definitions would include the vast array of clinical symptoms and not just the objective manifestations. I imagine you cover that as well in the chapter. Yep.
Absolutely.
Yep. And I'll just go over one more of hers before a few others that came in advance. At early prenatal office visits, the history of tick bite or past diagnosis of Lyme and other tick-borne diseases would surely ideally be included as well as part of health history, since testing is inaccurate sometimes.
Yes, I mean, I think that would be important. That would be part of the history. But also, it would be important to determine if someone who had a prior pre-pregnancy diagnosis of Lyme disease or suspected Lyme disease carried forward any sequelae into the pregnancy.
Yep.
So that population would be identified and included.
Yes, and another question is whether there's much resistance to this type of research that you've detected already, or not really.
Well,
I think
Laura,
I
you
was
want
going to
to
say, I
start
think
with that one?
I think that there is, at least in the US, and I'm assuming it's not unique, there is a real push to look at Lyme and other vector-borne, tick-borne diseases. We have a large national initiative that brings in patients, patient advocates, alternative ways of looking at Lyme. So I do think that this is a time that there will be much more acceptance and desire to do studies like this. But as Dr. Nades pointed out, the pregnant population is a challenging one when it comes to any kind of clinical study. But I do think it's an environment very interested in addressing Lyme and other tick-borne diseases.
Yeah. I mean, one of the advantages might be that in doing these kinds of studies with non-pregnant participants, there's always the issue of other remedies, whether herbal or pharmaceutical, that they're taking, and asking them to come off them for a longer period in order to have a sort of standardized approach. But with pregnancy, they're less likely to be taking a lot of different supplements, even if they do feel they have Lyme or other infections, wouldn't you say?
Well, if you have a large enough subject set, there will be individuals who will be taking supplements. There'll be individuals who are not taking supplements. And if you have enough people, enough subjects,
Mm-hmm.
then you can actually look to see if the statistically, you could look and see if taking the supplements has an impact on
Oh, that
any of
would
the
be interesting
findings.
too. There aren't
So
enough
that
studies.
would be one of the sub-analyses, if you will, within the cohort. Look at medication,
Mhm.
because also, we would be calling medical histories and looking at medication
Mhm.
histories, and what, during pregnancy, if someone was diagnosed with Lyme disease, were they being treated, what the outcomes were. So
Yeah.
if the numbers are large enough, you can make those comparisons and
Right.
answer the question, does supplements have an impact?
Mm-hmm.
And if I may just add about what was mentioned a few statements ago about this is now the time. I think more and more interest is being given towards gestational Lyme disease and because there are there are like the Cohen Foundation actually funding a lot of these initiatives in the United States. There's other foundations here in Europe that are looking towards funding these types of projects. The European Commission doesn't have one very specifically for this, but they are seeing, or they're showing interest in chronic Lyme disease, so
Yeah.
so I think that this is the time. It wasn't before, like 5 years ago, 10 years ago, but I think now the time is turning, and I think that these types of projects more and more are coming out, and I think that adds value to these two chapters, and the timing is really right. That it can be utilized for not only like to investigate, pregnancy in Lyme, but also pregnancy in other diseases, as Dr. Nates has mentioned. So the timing is right.
Yeah, absolutely. Another question here is whether you feel you'll be able to find sufficient chronic Lyme knowledgeable obstetricians
Mm.
and gynecologists.
Well, one of the advantages of the study design is that when we'd recruit a practice, we'll be agnostic to whether or not they believe that there is chronic Lyme
Okay.
disease. We'll
Mhm.
be looking at all their patients, and um, whether or not they believe there is chronic Lyme disease, we're still going to gather the data, um, and we'll be able to see, actually, we can always question the providers. Do you believe that there is such a thing as chronic Lyme disease? And then ask if their practices are different and the outcomes are different.
Right.
So again, with a large enough cohort, you can delve into the granularity of these questions and ask, you know, if you believe in chronic Lyme disease, that it's a diagnosis. Did you do anything differently for this patient? What did you do? And compare them to other providers who have been enrolled to see if, those who don't, do they do things differently, and are the outcomes different? So
Absolutely.
again, it's a
Yeah.
test of provider practice.
Thank you. Yes, I'll go on to a few questions that were asked in advance. What do you feel might be the long-term impact of establishing a gestational Lyme disease biobank for future research?
I think Dr. Gilliman could think of probably a number of things she'd love to do if she had access. Certainly, this would allow us to understand better the pathogenesis of Lyme disease infection
Mhm.
during pregnancy, both for the mother and for the fetus. Look at long-term outcomes. Plus, as I mentioned, we could look at other pathogens as well with this
Mhm.
resource,
Mhm.
and ultimately think about strategies for developing better diagnostic tools think of strategies for better therapeutic approaches, and ultimately know if if someone's in an endemic area and they're pregnant, do we need to be concerned? What is their risk? What is the risk to the mother? What is the risk to the fetus? What precautions might we take? It's a little bit like telling a smoker that they're at risk of lung cancer, and then when there's a spot on the lung that turns out to be benign, they're a little more incentivized to stop smoking. So the question would be, if you had if we knew that there was risk to pregnant women and their fetuses, would they change their behavior so that during that period they don't go deer hunting, or that period they don't go hiking in the woods? So there are potential impacts from this kind of study that are on multiple levels.
Oh, thank you. Yes, there was another question. How would you anticipate this research influencing public health policy or clinical practice? But I think you've included those answers in what you've just said, really.
But also knowing what the risk is, it may change provider practice in terms of surveillance during pregnancy, and we may find that there are certain things that one needs to do to monitor the fetus in terms of, are they infected or not, or or outcomes. So I think at all levels that is very impactful.
Thank you. Another participant is asking here, where would you find controls? Most areas where we think we have a non-endemic area turns out to have a lack of doctors who can accurately diagnose Lyme. Do you see that being a little bit of an issue?
Well, if you recruit an entire practice in a non-endemic area, then we'll have the ability, we'll have the history, we'll have the exam, we'll have the testing results in the laboratory. And so, whether or not the physician thinks the patient had Lyme, we'll have evidence to help support that decision, whether they had Lyme or they didn't have Lyme, at least based on the criteria that we have. So we avoid the selection bias of someone a provider being able to identify whether the patient has Lyme, because we're going to take all their patients, all the ones that are willing to participate.
Yeah. And I'll read a rather long one here that I've seen. Although I understand that in a project like this, you consider it most appropriate to use the tests that are currently generally accepted for a diagnosis of Lyme disease, in the name of progress, would it not be interesting to, perhaps in parallel, use some of the other tests that you've mentioned that are perhaps able to provide earlier detection and perhaps a greater chance of detection?
Absolutely.
Would that be possible, or would that be cost prohibitive?
Well, in this study, we decided there was no limit to our cost. And then through partnerships, but I absolutely think one of the big values of a study like this would be to gain that scientific and clinical data to support novel testing. That is where we have a gap. There are novel tests that are not standard in diagnostic clinical labs, and that's because there's not sufficient data to support them. And so could we generate sufficient data either with methods that are already out there but not used in common practice, or to identify new methods? So absolutely, I think that would be a key component
would be fascinating, wouldn't it? Maybe in parallel, so that you were standardizing to a certain extent.
One of the barriers to validating such novel testing is the cost of going out and getting clinically characterized patient material. We would already have that, so that barrier would be gone. So we would just need good science with the potential of providing a novel diagnostic, and that would be made available to the community after appropriate scientific review.
Well, I'll just add on one last question here, and then further questions have come in, so I'll review those and then send them on to you, if that's all right, Leona, for you to sort of send them in various directions, because I've got quite a few here that I haven't had a chance to go through that came in earlier. But one therapist is just mentioning that herbal therapies do sometimes help get the tissue pathogens out of tissues into the blood to be detected in labs. Otherwise, the labs may appear negative when in fact they're positive. So that's quite interesting, isn't it? But it's nothing that you'd want to do intentionally in pregnant patients. So I just think that's an interesting comment to have made. It is something that many doctors do mention.
Well, again, you know, if you have a large enough numbers and there are women who are using herbals who have been shown to be infected, we could then in the laboratory see, well, you know, in those individuals using herbals, do we find a higher rate of detection in blood? You know, that would be a very interesting observation.
Fantastic. Well, I do hope that we can continue these exchanges. Hopefully, soon enough, we will be actually talking about this sort of blue skies study that you're discussing. I'm sure that having this up will help to rouse interest as well. It's been really, really fascinating. I'm just coming off the chat and the Q&A in order to be able to see you again. I'm conscious of your time, so it is already just past 8. I just wanted to mention briefly that we're privileged to have Professor Gilbert again on the 9th of October with the authors of Chapter 6, who consist of, well, she'll be with her colleague Kunal, who I think you know, Mr. Kunal Garg, and also staff and directors from Armin Labs, who wrote that chapter. So that's Chapter 6 if you want to get the book, or you can access these chapters from the web as well, individually. So there are others after that, which will be going up on our website. We did mention the one on the mitochondria on the 22nd of October, but that's not yet up. So thank you so much again, our fantastic presenters today. Very, very interesting. And I'm sure there'll be lots of comebacks. So we will save over the chat. And Leona, I'll send you anything that we haven't managed to cover today. Is that okay?
Yeah, that's fine. Absolutely.
And thank you, everybody, for participating this evening. Thank you for taking the time.
Thank
You're welcome.
you.
Bye-bye.
Thank you.
Thanks a lot.
Bye-bye.
Bye.